It's my pleasure to introduce Antonio Costa, today's seminar speaker. Antonio is a physicist. He obtained his PhD from the University of Amsterdam, working with Greg Stevens. His work was very original during his PhD. He started introducing frameworks based on dynamical systems to characterize animal behavior. And his work has expanded from there. Today we're gonna hear about the latest developments. And thank you for being here.
Thank you, Federica. Yeah, it's a great pleasure to be here. Thank you guys for coming in a windy day. So as Federica said, I am indeed a physicist. I've been mostly working in the interface between statistical mechanics and nonlinear dynamics. But for already a decade now, I've been really fascinated by the interplay of structure and diversity that we see in how animals move, and how that reflects also a wide range of biological mechanisms acting across multiple scales, right? From motor neurons driving the motion of single muscles, to the coordination of the muscles by spinal networks, to then the modulation of these networks by neuromodulatory molecules, and how this kind of ultimately gives rise to how we move, right? And behavior is really also the scale on which evolution plays a key role, right? Variation in how animals search, smooth, and find predators, or find predators, avoid predators, and find mates, provides a substrate for selection. And so studying behavior, quantifying it in a high level of detail, offers an interesting lens for thinking about a broad range of biological mechanisms. But if that doesn't convince you, I'll jump into a more concrete example of where studying or quantifying behavioral variation can be directly relevant. And that's the case of neurodegenerative diseases. So here I'm focusing on Parkinson's, which most of you must be familiar with. And we know that it results essentially from the depletion of a specific class of neurons called dopaminergic neurons. And then this leads to a wide range of locomotor defects like tremors, slowness of movement, etc. And accordingly, treating patients with dopamine agonists to compensate for this degeneration can alleviate their symptoms. But one major issue that clinicians face is the fact that in a high percentage of people, around 40%, they can either not respond well to the treatments or develop complex side effects. And part of the reason for this is the fact that not only is the disease kind of mechanisms complex, but also this degeneration is acting on brains that are to some extent unique from one individual to another. And so the result of this degeneration is going to have a different impact. And so by quantifying behavioral variation in detail prior to these treatments, which reflects basically how these patients' brains are different from each other, we might be able to better resolve why different patients respond differently to different treatments, and so provide a more personalized response. And so I hope that I've convinced you that there seems to be a lot to be learned by thinking about behavioral variation in detail. Yes?
What extent is the response to treatment genetically mediated?
So the vast majority of Parkinson's patients are not genetically encoded. So it's sporadic cases that come from mostly environmental factors. So very little of it is genetically encoded. Yes, it's a really small percentage. I'm not a specialist, but I think less than 10% of cases have a gene identified to them. So yeah, maybe I'll get back to this example later on because this is something that actually we've been working on. Depending on time, I might come back to this example. But so, and thanks for the question, and please interrupt at any time if something doesn't make sense. All right, so quantifying behavior and especially behavioral variability seems to be powerful, but how do we actually do it, right? So these days, mostly due to advances in experimental approaches and machine vision, we can have a really high level of detail in our measurements of animal motion, capturing a broad range of spatiotemporal scales, right? From the fine scale movements of each limb of an animal to how these movements then get combined over time, for example, when searching for food, to even longer time scale circadian cycles and even developmental long measurements that have been done, for example, in the C. elegans worm. And so I won't get into the details of any of these different implementations. The point is that we can have these days like really precise measurements of the broad range of motions that animals can do across a broad range of spatiotemporal scales. And so we have this rich high dimensional data, how do we use this then to kind of make sense of variability in animal behavior? So a common approach to this problem is to essentially take steady state properties of the behavior, like, for example, the average speed of an individual against another, or the probability of visiting different kinds of behaviors. And while these kind of descriptions have been really valuable for behavioral ecology and our understanding of the structure to this variation, they kind of miss the fact that behavior also unfolds dynamically across multiple timescales, right? So two individuals might arrive at the same average speed, but one fluctuates a lot and the other one just stays there for the whole time. And also, for example, two mice here might have a similar bias for exploring and searching, but then once we look at a finer scale, they might explore or search in slightly different ways. And so if we want to have a more robust understanding of inter-individual differences in behavior, we really need to encode this kind of dynamic and multi-scale nature of behavior so that we can not have to make assumptions about what matters for the animal, but sort of allow the structure in the data to teach us about on what scales are these variations emerging and how that reflects underlying control mechanisms as well. All right, so this is kind of the broad problem that I'm now kind of going to try to formalize. So I like to think about these behavioral recordings as some observation of an unknown dynamical system, where this unknown state x of t basically is the entire set of variables that allows me to make a prediction of the future state of the animal. And so I'm assuming that this latent variable x evolves according to some first order dynamics, where we don't know what this function is, we don't know what these parameters are, and in most cases we also don't know what the x of t is. So I'm going to get into some of that in a minute. And so the idea is that basically variation among these parameters, for example, might encode how an individual might be different from another and how it behaves. And so basically what we are trying to do here is to kind of solve this inverse problem where we have these time series observations. We don't know what's the parametric control that is giving rise to the variation in them. And what we're trying to do is to basically reconstruct sort of the space of parametric variation and figure out, for example, if there are different types of like phenotypic structures that appear in the space of behaviors. All right, so but how do we do this? I mean, there are several challenges, right? If you just were to, let's say, compare directly, a naive distance among parameters, it can be very misleading, right? Especially in the context of having bifurcation points where, if you change the parameter by the same amount close or far away from a bifurcation, you have a very different macroscopic sort of change in the dynamics. We're also working from finite samples. So we need to have some way of assessing, how significantly different are two observations from each other. Could they come from the same dynamical model and just two finite observations of the same dynamics? So this is something that we want to take into account. And finally, we also want some metric that takes into account the fact that these dynamics are unfolding across multiple timescales. And so we need our distances to be sensitive not only to sort of the fast timescale properties of the dynamics, but also, more longer timescale statistics of, let's say, transitions between coarse-grained behaviors and things like that. All right, so to try to encapsulate all of these challenges, I took some inspiration from approaches in molecular dynamics and fluids. And instead of working with the nonlinear dynamics of the state, so the nonlinear evolution of the state, I decided to work with the evolution of probability distributions in the state space. I'll give you some concrete examples for what this operator P of tau is. But the idea is that basically we can use this representation to trade our nonlinear evolution of states to a linear evolution now of probability distributions, now revolving entire functions. But using this, we can then, on the one hand, try to make predictions about the future state by essentially sampling from the evolved probability densities. But also, because we're now working with a linear operator and we like to diagonalize things, we can look at their eigenvalues and eigenvectors. And it turns out that by construction, the eigenvalues of these operators capture different timescales. And the eigenvectors essentially encode for global structures of the dynamics that relax to the steady state on different timescales. And so the idea here is that we can use these eigenvalue and eigenvector pairs to identify or sort of separate long-lived behavioral structures, so long-lived sequences of movements, from the faster timescale pulse dynamics that we can measure in our data.
Antonio, should
Yes.
you be holding in mind that there's like a shadow of a Bayesian prior somewhere in this formulation, or is like where does the role of that type of thinking knit with this type of thinking?
Bayesian prior. I mean, you can think about this from a Bayesian perspective, but
You don't need to, you're saying.
I don't think you have to. No. No, I mean, I'm going to show you an example where we actually know what these operators look like, and then maybe we can come back to that question. Yes.
Is this related to Koopman?
It is the adjoint to the Koopman operator. Yes, it's the Proudhon-Frobenius operator. That's a good question. Let me walk you through an example, and then we can come back. So, this is an oversimplification, but let's imagine that the behavior of the animal is some kind of stochastic process on a landscape where the different wells might correspond to different stereotype behaviors. And I'm going to come back to this notion of what I'm calling a behavior, because the way in which I'm thinking about this problem is through the notion of a timescale separation, the fact that the animal does something for some time, that transitions and does something for another amount of time. And that you can think of it more or less, if you map it down to a potential landscape, let's hope the building doesn't fall, as some kind of complex landscape with different wells, where the barrier heights and the temperature essentially set the transition rates between different behaviors. So it's a very simplified description. But the reason why I'm working with this description is that you might be familiar with the Fokker-Planck equation, where you can now see basically where this P of tau is coming from in this case, right? It's just a discrete time evolution coming from this infinitesimal differential operator, which is the Fokker-Planck operator. All right, and so the cool thing is that we can, I'll show you in a second, we can infer these operators relatively well from time series data, and then we can use them to effectively decompose our dynamics across multiple timescales. And so in this case, besides the trivial eigenvalue, which corresponds to the steady state distribution, which is going to be the Boltzmann distribution, then we have a longest timescale that corresponds to the highest energy barrier, a second one that corresponds to the smallest barrier, and then a whole spectrum of modes that corresponds to the harmonic relaxation to each of the wells. You might remember your quantum mechanics classes, right? So it's equivalent to thinking about, actually you can map this onto a Schrodinger operator and then think about it in the same way. And so, as I said, basically, one of the modes, which is the mode that corresponds to an eigenvalue of P of tau of 1, meaning that if you act on it with P of tau, you don't move, you don't change. So that's the steady state distribution. In this case, it's just the Boltzmann distribution. And then the remaining long-lived eigenvectors essentially allow us to identify these energy barriers. So this is basically what I'm showing you here on the right, where you can see, this is a very simplified example, that roughly the sign structure to the first two non-trivial eigenvectors effectively allows us to identify where these energy barriers are in the state space. And so we can use this in general to build coarse-grained descriptions of our dynamics on different timescales.
Can you expand it?
Yes.
I got it. Like, why is it sufficient to look at the signs to get?
In this specific example, actually turns out that the signs are enough. In general, what you should do actually, so you can, basically what you're trying to do is to identify a set or a region in your space that remains coherent after some time evolution. And so you can directly measure that actually. So using P of tau, for example, you can arbitrarily define a set and then calculate basically how much of the probability is leaking out of this set. And then you try to find the boundary where basically most of the measure remains coherent after some time tau. It just turns out that it's, yes, another question.
Sorry, please finish your sentence.
Yeah, no, so it just turns out that in this kind of pedagogical example, directly from the sign structure of the eigenvectors, you get that. But also it's true that the sign structure also in some ways encodes basically, the slow modes of the dynamics, in the sense that 2 points that have a similar value of the eigenvector means that they communicate with each other in a fast time scale. Whereas if things are very far away in this eigenvector, it means that it takes a very long time before you reach the other side. So you're really sort of capturing the slow modes of the dynamics. Yes.
It says here you're explicitly talking about like a stochastic process. Where's the noise in your previous equations? Is it in the measurement equation or the latent variable equation? It's
in the latent variable equation. You can add some noise as well at the observation scale, but it's non-dynamical. I think it's more interesting the noise that you put in the stochastic process. Yes.
Does the neural system have to have a potential?
No. So I'm using a very simplified example, but all of this construction generalizes to dissipative systems as well. It's just I'm showing an example where we actually know, because it's a simple system of gradient dynamics. We know where the boundaries are. So I can show you that this construction recovers those boundaries. But you can use this as well in deterministic chaos or any other kind of dynamical system. All right. The point that I was trying to make here is that we can basically use the structure to these eigenvectors to effectively perform a top-down cross-graining of our animal's behavior, let's say. For example, using the slowest eigenvector, we can identify the first energy barrier, which is where this blue line changes sign. And then using a combination of the first two, we can identify the second energy barrier. And we can use this to effectively write down, on the one hand, kind of a reduced order model. But if you want in a coarse limit, you can even write down a two-state Markov chain that basically captures the transitions between being on the right-hand side versus being on the left-hand side of the state space, because there's such a stark timescale separation between that large energy barrier and everything else, that effectively you can write a two-state model and those macroscopic dynamics are preserved. Or if you want, you can add a third state using the second eigenvector and so on and so forth. And so basically, in the end of the day, what we are doing is that we're going from a fine scale, in some ways, a fine scale discretization of our space into a hierarchy of Markov models encoded by this parameter here, Q, which is telling us about the number of coarse-grained states. So Q equals 2 is a two-state Markov chain, 3 a three-state Markov chain, and so on and so forth. And so we're going at a decomposition of the dynamics from the longest timescales down to the fastest timescales.
At the level of data, you retain the first K modes that you infer carefully? Is that?
Yeah, at the level of data, you try to find an eigenvalue gap, typically. You can also try to estimate the noise floor by shuffling your data and then seeing basically what is the eigenvalue that you would get if you have no dynamics, and that can help you identify basically or separate the bulk of the spectrum from the eigenvalues that you can clearly distinguish. Yes.
So this coarse graining is purely based on where I am in X. There's nothing about dynamics.
No, it is to do with the dynamics because basically what you're trying to find are regions that communicate the least. So if you were to naively, I don't know how it would work, but I don't know, if you were to naively split in X, maybe you would split it in the middle. But you're not, you're splitting it here, which is the dynamical barrier. So it's really, it's a coarse graining based on the structure of the dynamics. Yeah. Other questions here? All right, let me show you how we can then use this thing to think about, this inverse problem of extracting variation from time series, okay?
So
yes.
can you elaborate on what the PIJs mean again?
Sorry, can you repeat?
Can you elaborate on what the PIJs mean?
So the PIJ is a discrete, approximation of the P of tau that I was talking about here before. So it's basically a discrete space approximation of this operator. It's basically a transition matrix, actually, a large Markov chain with many, many states.
So that's how you approximate this p tau.
Yes.
I see.
Yes. I mean, that's one way to approximate it. There are other ways. You can use kernel approaches as well. But the simplest way is just you partition your state space into small boxes and effectively you count the transitions, probabilities. It's very simple. Yes.
And so the idea in this simplified example is you simulate your dynamics and then try to recover the structure of the space from the data.
Yeah, so
that's what you'll have to do.
Exactly. So, I mean, the point is to illustrate here the challenge of, imagine that it's not this process, it's some random video of an animal. And basically, I'm going to try to play the same game later on.
Yeah, so from the data. Not
from the data, yes. Because here, the point of using this example is that we know what the solutions are. So I'm just showcasing the fact that we can recover those things without knowing about the theory, just by using the structure of these operators that we can infer from the time series. So from here on, what I'm showing is all done from simulations. So what I'm doing now is that basically I'm assuming or imposing actually that there are 4 different parameter regimes in which basically what we're going to vary is the depth of each of the minima. And so you have these 2 classes like orange and blue that share the first well, but then have different depths on the wells on the right-hand side. You have these 2 classes, green and red, that share again the first well, but have differences on the right-hand side. And so then we can sample, time series observations from these different parameters. And the goal is to now try to learn the fact that there are these different parameter regimes. All right. And so what we will do is basically what I was just describing before, which is that we deploy this kind of top-down coarse graining of the dynamics. We build this hierarchy of Markov models for different coarse graining scales. And then we define this metric, which is basically just, the L1 distance among the rows of this matrix. This factor of half is just to make the distance bound between 0 and 1. And so then we can estimate these distances across different coarse graining scales. As you'll see, this is really informative about the multiscale structure to the dynamics and the variation in the dynamics, right? So if I start with just a two-state Markov chain, meaning that I kind of erase information about the variation here, then I can only distinguish differences between, these two classes. So the orange and blue and the red and green, because they basically share the same structure on the left-hand side well, and we're destroying the information on the right-hand side. But if you go deeper into a three-state description, now we can distinguish all the different four classes. And so by looking at basically how these distances change across coarse-graining scales, we're getting information about sort of, the timescales that are most relevant for distinguishing different observations. All right, and so basically, using this, we can essentially measure these distances among coarse-graining Markov chains, which we can, abstractly think of as, living on some kind of embedded space where each point corresponds to one of these time series observations. The distance between these points is the distances that I was showing you before. But, there's also the fact that we're working from finite time series. And so there's some, inherent level of uncertainty about the exact position of these points in space. And so basically to try to assess this, we, design some kind of Monte Carlo approach to basically test whether 2 points could have been generated by the same underlying dynamical model. And so this gives us some kind of, effective uncertainty epsilon. And then we can look at basically how these two distances change as a function of coarse graining scale. And so as you can see here on the right-hand side, so the orange is the epsilon scale, the blue is the real distances that we estimate. We've shown that, and you can kind of intuitively understand that as you increase the coarse graining scales, effectively these two curves will collapse because we don't have infinite data, and so eventually we cannot distinguish significant differences. And so that explains the large scale behavior. You can also see that there's a difference between 2 and 3, which makes sense because we're at 3, we're unfolding this extra level of variation, and then basically this becomes flat. And so we can use this to effectively define a scalar sort of multi-scale distance among these dynamical observations by effectively integrating this region here. So taking the ratio -1, which goes to 0 at q into infinity, it's bounded. And so we can effectively just sum over all the coarse graining scales and get one value that encapsulates basically, how two observations are different from each other across all these different timescales.
Why is uncertainty increasing when you retain more modes? Sorry.
Because basically the more states you have, the more data you need to fit them properly. And so the uncertainty is going higher. Yeah.
Interesting.
It just scales with the number of states. Yeah. Yes.
Just to make sure I understand, in the Monte Carlo procedure, you're generating new samples from the inferred dynamics?
Yes. So what I do, no, it's not from the inferred dynamics. So basically what I do is that I collect pairs of points separated by a time tau from both trajectories. And then I use that to effectively generate a new transition matrix, Pij, that takes into account samples from both. And then I use the distance between that distance matrix and each of the ones that were independently estimated for each of the recordings.
It's sort of.
It's a bootstrapping. Yeah. It's an effectively a bootstrapping approach. Yeah. All right. So bottom line here, and we'll go back to behavior soon, is that we can get this scalar multiscale distance metric and then use that to uncover through using, for example, a diffusion map algorithm. I'm not going to get into the details of that, but figure out that there's basically two major axes of variation and that there are these 4 distinct groups of phenotypes. Yes?
For all this to work, right, I need very high sampling frequency, right? Like if I have in time, right, I don't observe very often.
So the sampling frequency will determine how well you can distinguish fine scale states. If your sampling frequency is fast enough compared to the highest energy barrier, then you will have an appropriate measurement of the 2-state Markov chain. But if already, you're effectively at the time scale of transitions between the two other wells, then you won't be able to distinguish that. And that will show up here, where essentially, basically, these two curves will collapse much faster. So it's really also taking into account basically, the sampling that you have of your data and how well can you significantly distinguish. So yes, it will depend on sampling rates, but it's kind of all encoded into how we're constructing the problem. Yes.
I just want to make sure I understand. A state is a type of motion, a particular behavioral motion.
Could be, yeah.
Do you also get the transitions between states? Because what follows what is probably of interest as well.
Yeah, you get the transitions between states.
Okay.
At different scales. Yeah. All right. So going back here, we see that, even though there was in principle 3 different parameters varying, actually they depend on each other. And so effectively you're living on a plane. And that's what we find when we look at the structure of that multiscale distance matrix is that basically there's 2 dimensions of variation and 4 phenotypes. Okay, any questions on this? I'll jump onto a real example of behavior then so that we can, maybe resolve some of these questions. Okay, so the original inspiration actually for thinking about this problem came from working with Claire Villard at the Paris Brain Institute. And this was mostly driven by the efforts of Gautam, who was a grad student in the lab. He's now at Okinawa. Gautam was really interested in this question of variation among individuals from the perspective of trying to understand basically how much an external environment sort of drives the behavior of an animal, how much of it is also left for its internal needs, if it's hungry, if it's stressed, etc. And so we worked on the zebrafish, which you might know as an established model organism in biology, particularly also in neuroscience because of its transparent head. So we can see directly the neural activity. And this is a lot what the lab of Claire does. And so these fish, they move through these kind of discrete bursts of tail motion. I don't think I have a video. That's a shame. Sorry about that. But you can believe me and you can look at the time series. Basically, they do these oscillations and they stop for a while and they do not stop for a while, etc. In this experiments here, this was done in collaboration with the team of Michael Orgard and João Marques in the Champalimau Institute in Lisbon. They had exposed the fish to different kinds of sensory contexts. There were about 14 or 15 different sensory contexts. So some of them were just freely swimming in the light, others were in darkness. Others had this kind of looming stimuli that emulates an approaching predator. Others were hunting for different kinds of prey. So there's a whole range of different contexts. And we essentially wanted to understand basically, how much of this variation comes from the fact that they are living or, experiencing different kinds of sensory environment. And so the data that we got from João consists of these sequences of bouts, where basically each bout, so each observation here is represented by this 8 cross 175. So it's 8 tail angles, 175 frames, which is roughly the duration of the bout. And what João gave us is basically this sequence of bouts.
2D.
In 2D, yeah. So they're swimming in a very shallow arena. They're also 4 millimeters long. These fish, they're quite small. At least at this stage, they're 6 days old. Yes.
In the experiment, are they in an individual state, or are they in a group?
Yeah, they are in an individual state. They're swimming in an arena by themselves. These questions about what would happen in groups is super interesting, but at this developmental stage, they're really young, so they don't exhibit much social behaviors yet. Think of this as like, I don't know, a 6-month-old baby, maybe one year-old baby. But they can already do really remarkable things. They can hunt, they can do all sorts of crazy stuff. I'll show you some of that. All right, so one challenge that we need to overcome here is that unlike the example that I was describing before of the dynamics on the landscape where I knew what the state was, here I only have access to these observations, which are some function of the state. And so I need to get some way of sort of recovering the underlying state. And so instead of directly naively trying to infer a Markov model from one bout to the next, what we will do is basically do a time delayed embedding, which means that we will build sequences of bouts and then try to determine basically how long a sequence do I need to narrow down my prediction of the future. So the intuition here is that basically if I only measure a single bout, I have very little information about what the animal is going to do in the future. But by looking further into the past, by looking into the longer timescale context, that can help me narrow down what the animal will do next. It's a little bit like in language, right? The meaning of a word, depends also on the context. And so instead of just directly naively trying to infer a Markov model for one bout to the next, which will lose some predictive power, we essentially expand our representation of the state through this delay vector x of t of k, where we look also at the past time delays. And then we try to determine basically how many of these delays do I need to maximize my predictability over the future. So we can quantify that by measuring the predictive information, where you can see that if you just take one bout, you're not doing a very good prediction. But as you stack them more and more, up to about 5 bouts, then you maximize your predictability over the future. Yes.
Why does
Finite size effects.
Okay.
Because basically the dimensionality of the space is expanding. These things become hard to estimate at large values. And so we're trying to get as much as we can from the finite data that we have. And in this case, this is what we get. Yes. There are situations where there might be longer timescale predictive information that we cannot resolve as it is right now.
So at the beginning of a bit, or sorry, a NAT or whatever, is what are the sort of over all 8 segments of the worm
of the fish?
Sorry, you can ask like 2 or 3 yes/no questions to determine what angle each. How would I determine?
Well, it's the whole angles as a function of time for an entire bout, actually. So we're looking at the distribution of these high-dimensional objects. Good. So it's not super intuitive to think about. It's basically, I guess, you could think about it in that way of how much information you have per bout, per unit of bout over the future. And I guess it's somewhere around 3.5 nats per bout.
Interesting.
Yeah. All right. And so basically, now that we have something that we think it's closer to a Markovian state, we can try to approximate this transfer operator in the way that I was describing before. So basically, doing a finite partitioning and counting. And then once we have that, we can use, again, the eigenvalues and eigenvectors to characterize behavior at different timescales. And so what we found, for example, is that, while most research in zebrafish focuses on statistics of single bouts, they actually seem to engage in long-lived sequences of bouts that can range up to a few minutes long timescales, which correspond to different kinds of, what we call these cruising and wandering states, where the cruising is mostly kind of smooth forward runs, and the wandering corresponds to combinations of different kinds of turns that it uses to do a bit more of a local search of the space. And so this is what we find at the longest timescales. I can give you a sense a little bit for what happens at slightly faster timescales. So there's also another second mode that roughly encodes for the speed of the animal. So here I'm plotting the 2 first modes against each other. And you can see the speed here in the color increasing. And the third mode that encodes for biases between swimming more towards the left or swimming more towards the right, which happens within a finite time horizon. But there's some persistence to their motion, left or right, and so on and so forth. And so the point is basically that we can now use these eigenvectors to obtain, this reduced order description of our behavior, whereby by defining sort of the values of these different eigenvectors, we can, qualitatively describe what the animal is doing while keeping all the fine scale information about, the structure of each bout. So without sort of imposing a coarse grading to begin with, but, looking at sort of, what emerges from the dynamics. Right, and so as we've seen before, we can then use this to do this coarse graining, right, at different scales. So here, for example, I'm showing you the 2-state Markov chain built between cruising and wandering states, which we can then infer for each fish. And what I'm showing you here on the right, just to give you a sense for how well we're encoding those dynamics, I'm showing you a result of simulations where what I'm plotting is the mean sequence length, so kind of like the average time spent in a particular state. So there's 2 states, cruising and wandering. So each fish has 2 points on this plot. On the x-axis is the data, y-axis is simulations. You can see that we're doing a pretty good job. And also that there's a broad range of timescales, right? From a few bouts to a few hundreds of bouts. Yes, question in the back.
Oh, sorry. Can we go back? Just 2 slides, just so. Yeah, so here. Can you elaborate again on the precise relation between the eigenvalues here and the timescale?
So the eigenvalues of this operator, so these eigenvalues basically relate to a timescale. Can I use the board? Is there? Maybe I can come back here. Actually, it's here. Sorry, it wasn't required. Actually, it's here. So you can see that if you do the discrete time evolution, these eigenvalues of L star, you can see them very clearly correspond to an inverse time. So it's a rate, right? If you diagonalize these eigenvalues. And so the eigenvalues of P of tau and are related to a time scale through basically this exponential form.
So the larger the eigenvalues, the slower the process.
Right.
Okay.
Exactly. The larger the eigenvalues of P of tau, or the smaller the eigenvalues of L star, because it's going to be a rate, right? So the smaller the eigenvalues of L star, or the larger eigenvalues of P of.
So ideally, in your experiment, you look at like the top 3 largest eigenvalues and dissect them.
Yeah, actually, we look at the whole spectrum. Here, I'm just showing you the first 3. There's other interpretable modes that we will actually use to eventually do this coarse graining of the behavior, which is what I was going to get at in the next slides. But is that clear?
Yeah, thank you.
These different eigenvalues are really sort of encoding different timescales. In the case of the dynamics on a landscape, basically that would correspond, for example, to the Kramer's rate of transitions across the highest energy barrier. That would roughly correspond to the first non-trivial eigenvalue. Especially if you're working a simple double well system, then it's a direct mapping actually to the first passage time problem across the barrier. All right. So basically, what I wanted to say here is that we can encode these dynamics, and now the point is that we can use also the remaining eigenvectors to build this kind of top-down description. I told you that the second eigenvector encodes something like speed. So at the 4-state level, we get also fast and slow variations of these cruising and wandering states. Further down, we get some left and right variations of these states, and so on, until basically we're down to the level of a single motor action. And so this is really cool because it basically allows us to directly sort of identify the behavioral traits or timescales that best capture inter-individual differences without having to make an assumption about what matters. Also because for every pairwise comparison, it might be that different timescales are more or less relevant. And so we want to have a way to basically encode all of these different timescales. And so, yes.
The label that you're offering for motor action, is that like you're saying that if you actually ask what the numerical value of that eigenvalue rate is, it's like of the stepping speed of a molecular motor, or what is that? How did you
molecular motor? No, I mean here it's the unit of the bout basically. Not the molecular motor. Okay. No, because we're working in the data. Our finest time scale is the bout level. Okay. So I cannot go below that.
Thank you.
It's good to clarify. Cool. Yes.
To ask about this idea of characterizing variability, are you repeating this procedure for each individual separately, or?
No, so basically I build an ensemble description across all the individuals, which builds my space of coarse-grained behaviors. Then once I have that space, then I infer these Markov chains for each individual independently. And as you will see, because of also this uncertainty scale, we will see that for some pairwise comparisons, we can resolve finer scales, and for others, we cannot resolve finer scales.
So it's the same state space, but different dynamics.
Exactly. So this just defines our coarse-grained states. And then, we can project each individual's trajectory onto these states, and then infer the Markov chains for each individual independently. Yes.
I'm wondering about how much these cruising and wandering describes like other cases where there might be predators or feeding or something.
Yeah, that's a really cool question, right? So right now I'm kind of assuming that the dynamics are autonomous. So I'm not taking into account, explicitly, time varying external stimuli. I'm basically marginalizing over the external stimuli, just saying, okay, overall the statistics of the behavior are these. That would be a more complicated problem because then you would have to add control, and the inverse problem would become a little bit more complicated. This is something that we've been thinking about, and if you want to discuss it more afterwards, I'd be happy to chat about it. But yeah, so right now, basically, I'm just thinking about the autonomous animal-centric perspective behavior of the animal, irrespective, or in some ways, I guess the behavior is encoding what the animal is seeing and what the external environment is, and we'll get to that in a second. It's kind of part of the question. Right, and so basically now, we can play the same game that I did with the triple well potential and measure the distance, the pairwise distance among every individual fish. So this is the whole distance matrix. There's a few hundred fish. So each of these rows and columns encodes basically a different recording and this kind of multiscale metric among different individuals. And so then we can, use that to effectively visualize or uncover sort of the major axes of variation among individuals. Here, each point basically encodes, all of the timescales of an individual fish, and the distance among them is how different their behaviors are across all of these different timescales. On top of that, remember that we also have this kind of notion of uncertainty, and we can explicitly use that to then group fish into classes, let's say, that we know are, statistically different from other classes of dynamics. And so the bottom line here is that basically, we can essentially organize these inter-individual differences along its major axis. So instead of just, assuming that this variation is noise to be averaged away, we're really trying to understand basically how the structure to this variation reflects underlying mechanisms. Yes.
Sorry, how did you cluster this? Could you explain it a bit?
Yes, I mean, it's going to take me a little bit on a tangent, but basically we have these distances, right? And we have these significances. You can map that onto a diffusion process with multiplicative noise because each pair of points has a different temperature, kind of. And so basically then you're doing a diffusion process on this manifold. And so then you're, again, basically doing a spectral clustering based on the eigenvectors and eigenvalues of the diffusion map. We're basically kind of playing the same game that we did for the dynamics, but now for the spatial structure on this manifold. Yeah.
And
no worries.
You can also play this game not only by dmax, but by d-resolved along certain modes. So,
absolutely. So you can ask the question, for example, if you have, here we're doing it in a completely agnostic way, so I'm not assuming anything about what's happening, but you can, in this case, for example, you could turn this into more like a classification task, for example, where you say, okay, I have these two sensory contexts. What is the timescale of the behavior that best distinguishes them? So right now I'm not doing that. I'm just sort of integrating over all the scales and then looking at what structure appears. But yeah, in principle, all this information you could keep. And right now I'm just summing over.
Do you see evidence that fish mostly vary on it's like, oh, two fish are totally different on these timescales, but totally similar on these other timescales?
Totally. Yeah, it's really cool. Actually, I don't have the figures. It's going to be on the paper. Also, by the way, all of this is kind of unpublished. So take some of it with a grain of salt. We're hoping to finish it soon. But indeed, the different kinds of pairwise comparisons, you will see structure appearing at different timescales in behavior, which I think is really cool. Also, for our neuroscience colleagues, they're very happy to see that. And so this is really exciting. But so, unfortunately here, we didn't have direct measurements of the underlying physiological variables driving this variation. But we could try to get a sense for what is driving this variation by looking at the average phenotype in each sensory context. So, sorry, this is a bit of a complex plot to look at. I'm going to try to walk you through it. So each of these tick marks here corresponds to the average in a particular environmental context. So the boxes here correspond to different conditions, like being in the light, different kinds of arenas, different types of stimuli. These triangles correspond more to kind of aversive-like stimuli. I'm not going to get into that. And these ticks on the right-hand side correspond to fish that were either exposed to prey or hunting during the experiment. So these are the ones that have, a little microorganism on the right-hand side. And the little tick mark on the left indicates whether these fish were also raised with prey during their past development, or whether this is the first time that they're seeing prey. So these two guys is the first time they're seeing prey. This guy here has seen prey in the past, but during the experiment, they're just freely exploring in the light. And an important aspect to keep in mind here, again, is that these fish are 6 days old. And so they carry a yolk sac. It's like they're carrying their eggs around. So they can feed themselves. They don't have to actually actively hunt. And so all of these fish here on the left have never seen prey. All of these fish here on the right have seen prey either during the experiment or during their development. And so that seems to be one of the major sort of, axes of variation among fish, which is whether they have never seen prey or whether they are either seeing prey during the experiment or have been raised with prey in the past and now are just freely exploring in the light. And so you can see that basically, these two fish, so the fish that are in this bottom corner and the fish, for example, that are here, both of them are seeing the same thing. They're freely exploring in the light. The only difference between them is that some of them were raised with live prey before and others weren't. And that makes a huge difference in how they explore space and how they navigate. We can pick that up. And so overall, it seems like, prey exposure and also past developmental histories can have a big impact on how the fish navigate space. But we also found that even within a particular sensory context, there's still quite a bit of variation among individuals. So for example, if we focus on one of the conditions where the fish is hunting for prey, they're hunting for paramecium. You can see that the vast majority of fish are on the right-hand side, as I was showing you before, but there's still some fish that are behaving closer to fish that have never seen prey in their lives. And what's cool is that the fish also have a bit of a tail when they try to hunt. So if you look at their eyes, they converge their eyes on the prey when they're trying to hunt. And so we can get a sense for how much they're actually trying to catch prey or not. And so when we do that, we actually find that the fish that were behaving as if there was no prey and they're doing this kind of broadal dispersal-like behavior, they're also not even trying to hunt. Whereas the fish that are here on the right-hand side doing more of this local search are the fish that also are actively trying to hunt. And so we can decisively say, what is driving this variation, but clearly there's something about the internal state of the animal that sort of overrides the fact that there's prey in the arena and they're behaving as if there is no prey. And so they go more on this left-hand side.
Would you use this
Right.
to learn, if a fish watches another fish and acquires a behavior, could you see that with this type of approach?
If the fish what, sorry?
Watches another fish.
Watches another fish and acquires, yeah, I guess you could try to see, how similar their behaviors are. Mimicry, yeah. At this stage, development, I don't think they do that, but later on, maybe, yeah, yes.
If you look at this longitudinally, as you go through different stages, what do you think? How would this change?
That's my grand proposal. This is what I'm going to be doing. Yes.
I would fund you because this is
this is what I'm going to be doing actually, where I'm setting up for the first time experiments that I've never done in my life to precisely track individuals throughout development and look basically how the structure to this variation changes on developmental timescales.
It'll dictate. It'll be a nice dictation too to see if it's environmental or if it's from
100%. Yeah, we need to keep track of the individuals on a longer time scale to be definitive about this. Yes. So this is why I was a little bit unsatisfied with these results. I mean, it kind of inspired us to think about this problem more broadly, but I also wanted to work on an example where we also can measure the underlying control mechanisms. And so I have time. I think I have 10 minutes. 5 minutes? 10? Okay. So, I mean, most of you will be familiar with the system, which is the E. coli bacteria. And basically, these bacteria move by essentially changing the rotation of their multiflagella, which drives them either to perform runs or tumbles. This has been very well characterized, and they basically can use or change their tumbling bias to effectively climb chemical gradients in their chemotaxis behavior. And now the cool thing is that the team of Thierry Monet at Yale has measured not only the movement of the bacteria, but also the expression of key chemotaxis proteins. So they designed these mutants that basically overexpress either key R or key B. So key R and key B are proteins that are involved in basically the adaptation of the chemotaxis network. So key R activity basically methylates the membrane receptors and increases the sensitivity of the network and makes the bacteria tumble a bit more. Whereas key B basically reduces the sensitivity of the network and enables the bacteria to adapt to different kinds of levels of chemicals. And so as you can see, basically, they genetically engineer these bacteria to either express key R or key B, and depending on the ratio between these two proteins, you see kind of different kinds of behavior and a broad distribution where if key B is highly expressed in comparison with key R, there's a low tumble bias, so the bacteria is mostly running. Whereas if the opposite is true, then there's a broad distribution of tumble biases, and the bacteria is tumbling a lot more. And so the cool thing here is that we have access to the behavior, and we also have access to these expression patterns. And so the question is, can we use the behavior to say something about the variation in expression patterns? And so basically, we're going to proceed as before. Now, instead of working with bouts, I'm working with the actual trajectories of these bacteria, which we can characterize in terms of perpendicular and parallel velocity components. Then we can play the same game that I was telling you before, do a time delayed embedding and build these Markov chains. And we find different types of modes. We recover back this kind of run and tumbling mode directly from the structure of the eigenvectors of the transition matrix, but also other fine scale modes that have been less characterized, like an intermediate speed state, more curvy or more directed runs and stuff like that, that we are not exactly sure how the bacteria controls for that, but it appears in the data. It could also be some boundary effect or something. So I need to talk a little bit with Thierry about this, because when they're close to the surface, then you have hydrodynamic effects that might drive these changes. But the bottom line is that basically now we take this large collection of bacteria, there's thousands of them, we play the same game as before. We build this hierarchy of Markov models and then measure how different each bacteria is from each other. This is the distance matrix. And then we can again sort of embed this into a lower dimensional space, where we basically find that there are two major axes of variation that roughly kind of correspond to changes in tumble bias. So if you're up here, you're tumbling a lot. If you're down here, you're not tumbling much. But now the cool thing is that besides these behavioral metrics, we also have the gene expression. And so we can look at the expression patterns of KR and KINB and see how they correspond to these recovered dimensions of variation. And what we find is that the first mode, so the macroscopic or the largest scale change in variation in the dynamics, is basically corresponds almost one-to-one to the KB concentration. So you can see here, this is color coding the KR and KB expression by the projection along this diffusion map eigenvector, where you can see basically as you go up, you increase in V1. Whereas the second mode kind of encodes for variation in KR, but only when KB is also high. And so this kind of highlights a bit of a hierarchical structure that exists in the chemotaxis. The fact that KR's activity kind of happens independently of what the bacteria is doing. So basically, they're just constantly methylating the membrane receptors. And so if there's no KB, if KB is low, then basically the bacteria will be always tumbling because the receptors are super sensitive because there's nothing to counteract that. So only when the KB expression is large enough, then you are within the dynamic range where KR actually makes an impact. And so we can neatly see that just by looking at the structure of behavioral variation in the bacteria. And so, using the behavioral alone, we've been able to basically recover these underlying control knobs and also sort of reflect interesting, subtle structures within the biochemical network that are, hard to see even just by looking at, the network itself. All right, and so maybe I'll wrap up here, or do I have time to 5 minutes? Okay, so maybe I'll just go back all the way to the Parkinson's example, or if you have maybe any other questions. No.
Can you resolve anything about timescales with this inference? So, do you see that, molecular things are different when expression of KR versus, KB? Do you see that extracted from your data?
Well, effectively, the timescales are encoded in the transition rates of the Markov chain. So the fact that this variation is popping up means that there is differences in those timescales.
Yeah. It'd be cool to see if it's consistent with the molecular understanding. I don't know what the molecular understanding would be, but yeah.
Yeah.
That's cool.
I'm not sure if we have that level of molecular understanding to know exactly sort of, how the entire network drives these timescales, depending on expression patterns.
People do the 2000 systems biology inference of the OD E parameters, however fraught that is.
Sure.
So, but in there's a rate associated with the induction of KR versus the induction. I don't know, when maybe it'd be fun to just do the comparison.
Yeah, that's true. Yeah, that for sure. We know each component basically what's the rates, but then at the level of the entire network, how that results in emergent timescales and how that maps onto the tumbling rates, I'm not sure. But yeah, it would be nice to look into it. If we want, we can do that together. Yes.
Back on a previous question about collective behavior, what is the setup here? Are they swimming together? And is there a patterning in this group based on their expression, their motility behavior?
I don't know. From what I understand, it's very low densities. So there isn't much collective behavior happening. Yeah, but it would be interesting to try to map how, these expression patterns and how they drive the behavior have an impact on the emergent collective behaviors if you put them at a higher density. But I think here they are at very low density, so there aren't much collective effects. All right, cool. So just to maybe wrap up quickly, if there aren't any other questions, I'll just quickly show you what we've been doing with humans. This is also a little bit preliminary, but so basically I've been collaborating with some neurosurgeons at the Paris Burn Institute that implant these deep brain stimulation electrodes in a particular region of the brain that has been used to lower the severity of Parkinsonian symptoms. And they've also collected this longitudinal data over a few dozen patients where they've measured how they're moving prior to the implant of these electrodes and also 6 months after these electrodes have been implanted and they've been under this deep brain stimulation strategy. And so here we have to simplify our analysis because the amount of data and the cleanliness of the data is not even comparable to what we can collect with fish, obviously. But we can play a similar game and try to understand basically whether we can predict which groups of patients will respond better or worse to the treatment just by looking at the behavior. And so it turns out that basically, some notion of coordination along the axial dimension between sort of trunk, pelvis, and lower limbs is a good predictor of basically, how each patient is going to respond to the treatment. In this specific symptom, which is called the freezing of gait, which is one of the most severe symptoms where they really cannot initiate movement and they typically fall and can, hurt themselves severely. And this happens still, in a vast percentage of patients, almost 50% of them keep on having these freezing of gait events. And so the goal here is to use these kinds of approaches to now sort of apply this in the clinical setting. And help decide whether it makes sense to do this complicated surgery to the patients or not. So just to give you a bit of a practical example of how these ideas can be relevant in every day. And so I'll just wrap up quickly. I'm not going to show you any of that stuff. Yes, so I've told you about how we can sort of use these transfer operators to think about the multiple timescales of behavior, and how we can then use that to study this multiscale variation in the dynamics and how it reflects internal states. I'm now, working together with Claire to try to understand, using the tools that we have in the fish, how these different axes of variation reflect changes in neuromodulation in the fish. And I also have this other ongoing project for which I'm hiring, of trying to understand how the structure to this behavioral variation reflects the unpredictability of natural environments where fish have evolved. And to do this, I need to do this kind of developmental level tracking of the emergent variation. And so there's also going to be some field work in different locations in the Indian subcontinent, collecting fish that have evolved in different kinds of environments, to try to understand how does that sort of determine the amount of inter-individual variation in behavior that helps the population cope with fluctuating. And so, yeah, with that, I'd like to thank you guys again for the invitation. It was really fun to be here, and we can, have more questions. Thank you.